We are now supposed to discuss certain things regarding basic implications of life sciences in biotechnology forum. That is industrial biotechnology. How many of you have heard this term? Industrial biotechnology, raise your hands. And rest of you have not heard the implication of life sciences. Industrial biotechnology is not a subject, is the core implication of life sciences. You can see here it looks very much theoretical. Am I right? It looks very much theoretical, but the implication of this image is huge because it is the key of the entire journey in the biotechnology. Irrespective of your interest in the specific domain, whether you want to pursue your career in biotechnology plant, biotechnology animal, biotechnology microbe, then eventually these are being termed as industrial microbiology. Industrial microbiology, right? So that you are understanding the implication of microbes for a particular cause. But the basic root cause or the basic, you can say the beginning of the entire implication journey starts with this gene. Whether you are taking the system of microbes, then where you will land with? That is very important question. You are taking the cell line of animal, where you will land with? That is very important. Ultimately, the pathways you are taking, you are following are different based on the system, animal model system, plant model system. What is the plant model system name? I have already told you earlier. Can you name that plant model system? Arabidopsis thaliana. You have not heard this name? Arabidopsis thaliana in the plant system. Similarly, in the animal model system, guinea piggies. Why these important two names are being taken in the industrial biotechnology? Because no country can afford the price unwanted unnecessarily without putting any mind. So on the experimental side, demonstration side, you need to establish your pilot project with this model system. This slide is also saying the same thing because we start with the homolog sequence. How many of you have heard this name, homolog sequence? Very good. What is the meaning of homolog sequence?
So their sequence are going to be similar.
Yes, representative sequence. In the another term, the representative sequence of the entire flora is very much captured in that domain, that is Arabidopsis thaliana. Similarly, the representative sequence of the entire fauna, you can say the animal systems, being captured in the model mouse system, आपने सुना होगा, animal system. So that whatsoever you want to do, whether if you are a clinical researcher, then of course you will go with this kind of the animal model system, that is guinea piggies, because we do follow the bioethics also. We are not supposed to sacrifice the serious, you can say model system like human body, like animal bodies. But the guinea piggies have been given the kind of the apathetic, kept in the apathetic category and sacrificing, right? So you will deal with them in due course of time when you will join the higher research in your coming, career. So that is why try to understand what is homolog reason. Being a biological scientist, you need to understand from which part of the genome we will begin with that. I'm giving you kind of question, you have to extract certain things. You have to extract certain things which is very much destined for the robust behavior against any against any toxic element. That is, of course, that is drug. You will talk about the antibiotic. You will talk about the specific drug. You will talk about any kind of the metabolite. So these are the several examples being known as, being understood as the ultimate outcome or ultimate product of the system being used in the biotechnological process, and ultimately with the help of the different technological implications at the industry level, people are harvesting certain things. Any example in the nature which is very much having the industrial implication or biotechnological implications in the natural yard? Have you heard this?
BT cotton.
BT cotton plant. It is an example. It is not meant for any drug delivery purpose. It is serving your purpose in the food basket, because BT brinjal has a different kind of the example. I don't want to bring this thing into this kind of the discussion when I will have the separate session on the BT cotton, which is very meaningful and very contemporary theme in the biotechnological world, right? Of course, has been owned by Monsanto, right? So these are also specific avenues, or you can say the destinations where you can explore your future. That is also very important thing. I was talking about the specific things where this particular logical thematic background can help you, how you will figure out any specific product with the help of the biotechnological tools and techniques. I was trying to name any medicinal plant, any other kind of the system which is very much important for the biotechnological implication. For example, take any medicinal plant, neem. Its biological or its botanical name is Azadirachta recta indica. You must be knowing n number of the products have been harvested with the help of this biotechnology tools and techniques. And of course, people who are sitting in the pharmaceutical research, drug designing research, clinical research are having broad spectrum knowledge at the molecular level and harvesting the things to the mankind. So that similarly, n number of hidden substances are there in the system of nature. You are supposed to unfurl it and then make them useful for the mankind. That is the basic purpose. That is why we are meant for, and being a life science student, we are supposed to understand these procedural things with the defined SOPs. Right? SOP का नाम तो सुना है? Standard operational procedure. That is meant for when you will, right now you are at the beginning side and you are understanding, being inducted to this biological research, and slowly you are also developing your incline, your behavior, your aptitude, so that you can also reach that process. Understand this homolog region. I was talking about any alpha, beta, gamma genome you take, which is very much meaningful. And of course, before naming any alpha, beta, gamma, you have to make sure, yes, nature has given certain gift of gab, which is very much meaningful for the existence of human being. Because we are selfish people. We are selfish people, and we are always trying to understand nature with our own terms and conditions. If nature is not fulfilling our terms and conditions, then we think this is the kind of poison, this is the venom, this is the kind of any kind of toxic elements, so we should be away from these things. Because the receptors are not available in our system, that is why that thing becomes unwanted, obnoxious. So this is another example of the nature in the biotechnological era. Okay, so that is why biotechnologists started to understand the things at the genome level, because genome is a huge pocket huge, you can say, stuff. It is not possible to figure out the entire genome at one go or in a few years. It takes years and years, decades and decades to decode the specific genome of XYZ. For example, I have taken the Arabidopsis thaliana, and if you have taken this Arabidopsis thaliana of this genome, for example, it's 10 kb base pair size of this genome. I'm not taking proper size of this Arabidopsis thaliana, I'm just exemplifying. If you are taking any genome in your account and trying to read it out, then it will take years and years, decades and decades. Because let me tell you, 99% of the genes, those are sitting here in a different manner, they are not useful. So that is the basic question for us, how to make them useful. Because you must be knowing nature and genus or the genome. You have heard the term genotype versus phenotype. So that phenotype is the external terms and conditions given by the nature, is very much perceived or imbibed by the genome, which is also the inside nature. If both of them are coming together, then making a meaningful dialogue and then giving you certain product. So that kind of the engagement process we need to learn being a biological student. Only then you can capture, you can figure out certain product at the end. So same thing through this slide, it is very fundamental slide, homologous region you have to find out from that genome of any given species, whether it is known or unknown. With the help of certain parameters, that is why we need to understand bioinformatics, tools and techniques. That is also very important. We need to understand the biostatic, you can say, simulatory pathways. That is also very important to figure out certain things, and then at the end you will find out this homolog region. It is very much readable with the help of the sequencing technology. Sequencer का नाम सुना होगा आप लोगों ने. Sanger sequencer is primarily, it was known as the Sanger sequence. Now we are in the third generation Illumina, giving you the digital capturing pathways. क्या बोलते हैं हम लोग उसको? कौन सा sequencing बोलते हैं? बोलो. Which sequencing? Pyrosequencing. Now we are in the pyrosequencing. Now we are in the third generation sequencing. So many next generation sequencing. There are so many terms and conditions to decode that complicated genome, right? Then, because it is not possible to read them unnecessarily at one go, we need to read them in a very simple manner, so that slowly you will unfurl at the chromosomal level which chromosome you are targeting, because these set of the genes are not very much specific at a site. Might be the sudden homologs, analogs, orthologs. You must be knowing these terms. Orthologs, homologs, analogs. Structurally, functionally and evolutionarily. You need to learn these things. That is very important. That is why these homolog region becomes very crucial for a biological student at the research level, so that now you are targeting the genes. How many genes are there for a particular purpose? For example, a gene is meant for n number of purposes. Another vice versa, certain purposes, several processes or several purposes are being dealt by the same gene. So we are supposed to understand with the different expressional pathway. That is why we need to take the help of the microbial domain, prokaryotic domain, where you can capture these expressional activities in a very simple manner, in a very quick time, you can say stipulated time of frame. Are you getting my point now? Am I clear saying this thing? That is why we are targeting the genes. Then again, you can say here the forward and the reverse, downstream, upstream, both of the things. It is a vice versa. And then this vector you have taken, this vector could be anything. You have to understand yak, back, yeast artificial chromosome, bacterial artificial chromosome. Nowadays, so many kind of the you can say vector types are there. Again, cloning vector will arrive in between. Destination vector, integrated vector, expression vectors. So you will read all these things in your upcoming sessions, or you can say the semesters, right? So that is why ultimately this recombination is possible. With the help of these things, the recombination, what is the meaning of recombinations? Means you have altered certain sequences in between based on the feasibility factors. Based on the feasibility factors, then at the end, you are expecting certain novel things from as compared to the primitive genotype. That is very much wild type. You must be knowing two types of the trait, wild type and the elite type. The recombinant version is always known as the elite type because it is solving or it is giving the or satisfying your purposes, right? Solving your purposes. And that is why people are very much dependent on this kind of the biotechnology that is meant for industrial implications. Now, question begins. Why we should, if gene is doing certain things on its own in the nature, then why we are supposed to squeeze it out unnecessarily? We are reading them unnecessarily. We are modifying them unnecessarily. And of course, somehow it is compromising the system. At the cost of the system, it is also, there are so many if and buts. A layman can ask you this question, huh? Because you are dealing with such a serious system that is very much biological. You are not dealing with the abiological system. Whether you are talking about prokaryote, talking about the eukaryote, everything is living system. And you are not, nature has not given you the authority to dictate on the natural terms and conditions unnecessarily. So what could be your answer as a biological student? Tell me.
We are studying or expecting on the genes because due to certain conditions, when unnatural conditions are favored by humans only, then we can expect or manipulate the certain genes.
Right. In another way, you can explain we are not fiddling with the natures. We are not destroying nature. Rather, we are mimicking the nature. We are mimicking the nature. We are establishing parallel lines of the nature. And we are trying to magnify the products. Because swelling population is multiplying by n. But the resources are being constant. That is why we are trying to understand the products and of course the meaningful products we are targeting. An example, I have taken you the example of biotechnology industry, Monsanto. Monsanto is an American company and it is very much establishing its venture in the Indian soil also, and it is doing a wonderful job with the BT cotton, BT brinjal, right? And some other. Similarly, Indian companies like Bharat Biotech, like Tata's, or you can say ITC, Cellulose Hub, Reliance Biotechnology. These are the peculiar Panacea Biotech. These are the important biotech ventures. Those are Zydus Cadila. Those are giving you opportunities for the employment with these processes. So if you are only acquainted with the theoretical things, not solving their purpose, you are supposed to be smart and giving certain, raising certain questions and solving on your own, and then extracting certain protocol and then being equipped for the industrial purposes. Only then they will hire you. Otherwise, there is no scope, neither in India nor in any other countries. Let me tell you. So, this concept is helpful for your basic acquaintances so that your IQ level will help it up to the certain extent, to bring it to the certain extent, and you need to understand the basic problems being faced by the society, and then you are coming out. Otherwise, biotechnology will always be floating, unlike other technologies, like you are enjoying electronics, you are enjoying other computer gadgets, because they are understanding and creating proximity with the societal problems, with the societal demand. Similarly, we life science people, we biological scientists need to understand the health related issues, the hygiene related issues, the ethics related issues. Okay? So this is the kind of the implication of this biotechnological or industrial biotechnology. So in conclusion, we are trying to draw, we have to clear our basic doubts. That is why we are traveling with a different semester process, and at the end we are reaching towards the implication of this particular curricula, that is life science, BSc, MSc integrated, and at the end we will be able to figure out certain kind of the title, whether you are in plant science or in microbial or in animal. Doesn't matter. Matter is that how brilliantly you are raising or you are bringing a questions in your mind, and then you are putting it before the forum and then giving the solutions also. Because only raising the question is not end of the story. You need to solve it with your acumen which has been taught in your classroom. So slowly